Hi, my name is Rajaiman and my matrix number is 180878. For this practical, we were assigned to do research on canine Gaucher's disease. So what is canine Gaucher's disease? It is a rare autosomal recessive genetic lysosomal storage disease which results from the dysfunctional metabolism of sphingolipid and also the accumulation of glucocerebroside in cells. So this is mainly caused by the defective activity of glucocerebrosidase, which is responsible to degrade the glucocerebroside. So this will result in the accumulation of glucocerebroside in most regions of certain organs such as the brain, liver, and the peripheral lymph node, as this was reported in an Australian Silky Terrier in year 2008. So what is exactly this Gaucher disease? Gaucher disease is caused by Gaucher cells, which are enlarged macrophages. Macrophages are white blood cells which contains the undigested glucocerebroside, which is due to the dysfunctional metabolism of sphingolipids. That is the reason why. So, what are the clinical signs from this canine Gaucher disease? So, the first would be anorexia, which is poor weight gain. There's lymphadenopathy, which means the lymph nodes are enlarged. And then there's general ataxia, which means there's lack of voluntary muscle movement or control. And they have a wide base, stiff gait for balance, and generalized trembling, and also hyperkinesis. So there are several ways to diagnose this particular disease. First would be the histology of brain sample, but this would normally be from a dead patient, and also we can check the cerebroside content of liver and the brain. So one of the ways is we could stain the brain by Laxol fast blue, followed by periodic acid sheath and this hematoxetin. So from there, we can diagnose it from under the microscope that we will we can observe there's this storage bodies of Gaucher cells, which looks like a tubular structure. So these are the abnormal amount of lipids in the cell called the glycosphingolipids stored in the reticuloendothelial cells, which ends up to be the Gaucher cells. So this would push off the nucleus aside from the cell and would fill the remaining space in the cell or the cytoplasm with lipids. So from there, that is when we could diagnose to see there's the presence of Gaucher cells. Okay, that is all from me. Thank you.
My name is Elin Sen. My magic number is 175282. Okay, today I would like to talk about the canine Gaucher disease. Okay, firstly, glucosylceramide is actually a glycolipid that contains fatty acid, glucose, and sphingosine. They're also called glucocerebrosides. Glucosylceramides are commonly found in animal cells such as spleen, erythrocyte, and also nervous tissue, especially neurons. The enzyme that catalyzes the breakdown of glucosylceramides into glucose and ceramide, which is the body used for energy, is called glucosylceramidase, found in lysosome. In this Gaucher disease, lack of enzyme glucosylceramidase will actually cause the accumulation of glucosylceramides in tissue macrophages through phagocytosis in spleen, forming Gaucher cells. These cells accumulate in perivascular spaces in the brain, bone marrow, liver, and make them unable to work properly. Following the accumulation of the glucosylceramides in the body, it will actually cause a spleen and liver enlargement and develop together with lung and bone problems. Okay, this is because when there is accumulation of Gaucher cell in the spleen will actually continuously break down the blood, and that faster than it could be produced. And this will lead to the lack of blood cell. In animals, the main Main sites for degradation of glucose phospholipids are the lysosomes that contain digestive enzyme. These enzymes are active in acidic condition. Saposin C is essential in degradation of glucosylceramides, but due to lack of this enzyme, there will be also more glucosylceramides produced due to the biodegradation of all red and white blood cell. They will also stored in lysosomes and interfere with their normal function. Thank you.
And my matrix number is 180864. Today my topic is about storage diseases. Group of genetic diseases associated with failure of metabolic pathway, which is the accumulation of substance within lysosomal system, such as lipid or protein within a tissue, which lead to the impaired cellular function. The two most commonly reported storage diseases in veterinary medicines are lysosomal and glycogen storage diseases. Canine goucher disease is one of the diseases that I will be discussed today. So goucher disease is a rare autosomal recessive genetic lysosomal storage disease of dogs characterized by dysfunctional metabolism of sphingolipids and accumulation of glucocerebrosidase within lysosome. Goucher disease is the most common lysosomal storage disorder and is caused by the defective activities of glucosylceramidase, also known as glucocerebrosidase. The lysosomal hydrolase that is responsible for the degradation of glucosylceramide. As a result, the autosomal recessive genetic defects those enzyme accumulation intracellularly. Tissue macrophage engulfed with glycolipids bundle lysosoma, known as Gaucher cells, are the hallmark of the disease. The condition has been reported in the Australia Silky Terrier and involve accumulation of glucocerebroside in most regions of the brain, livers and peripheral lymph nodes. Because the canine case involved the brain as well as peripheral neuropathy and rapid clinical deterioration, canine glycocerebrosidosis closely resembles human juvenile glycocerebrosidosis. Clinically affected dogs present at 6 to 8 months of age with poor weight gain. This is usually marked in coordinating with the general ectasia. Diagnosis is based on presenting neurological signs, MRI imaging of brain to exclude other differentiation neurological diseases, and histology of brain samples. Histological analysis is usually diagnostic with characteristic storage bodies in Gaucher cells containing tubular structure characterized by mild to moderate spongy vacuolation of white matter. DNA testing is currently unavailable for this disease in dogs. The condition is usually progresses for 4 to 6 months after the onset of clinical sign and is fatal. The younger the dog is when it begins to affect it, the more rapidly the sign progress. So in this experiment, there are a new word I have learned, which is ectasia, which is mean the loss of full control of bodily movement. So that's all from me. Thank you.
Today we will discuss on canine goucher disease. By the way, my name is Sim Yiling and metric number is 180893. Canine goucher disease can also be called as neurovisceral glucocerebrosidase storage disease. That is a kind of rare autosomal recessive genetic lysosomal storage disease of dogs characterized by dysfunctional metabolism of sphingolipid and accumulation of glucocerebroside within lysosomes. Gaucher disease is the most common lysosomal storage disease and is caused by the defective activity of glucosyl ceramidase, which is also known as glucocerebrosidase, a kind of lysosomal hydrolase that is responsible for the degradation of glucosylceramide. As a result, the autosomal recessive genetic defect, these enzymes accumulate intracellularly. Tissue macrophage engorged within glycolipid-laden lysosomes, known as Gaucher disease, are the hallmarks of the disease. The condition has been reported in Australian Silky Terrier and is involved in accumulation of cerebroside in most regions of the brain, liver, and peripheral lymph node. The clinical signs are poor weight gain, lymphadenopathy, and progressive neurological signs. And there is also usually marked in coordination with general ataxia, a white-based stiff gait, generalized trembling, hyperkinesis, hypermetria, exaggerated limb reflexes, but normal sensory reflexes. Histological analysis findings include characteristic storage bodies in culture cells containing tubular structures characterized by mild to moderate spongy vacuolation of white matter. The condition usually progresses for 4 to 6 months after onset of clinical signs and is fatal. The younger the dog it is, when it begins to be affected, the more rapidly the signs progresses. Due to the severity and lethal nature of this disease, affected dogs are often euthanized. That's all from me. Thank you.
Hello, my name is Siti Jasmina binti Sheikh Husainuddin, metric number 180880. So we were given a topic on storage diseases, a group of genetic diseases associated with failure of metabolic pathway. Because we were given canine Gaucher disease, group number 18. So storage diseases, they have an effect on cells, particularly the lysosomes, in a sense that there's an accumulation of substances within the lysosomes. And in canine Gaucher disease in particular, there's an accumulation of glucocerebroside. This canine Gaucher disease, it's a neurovisceral glucocerebroside storage disease. By neurovisceral, I mean the innervation of the internal organs by the autonomic nervous system. And hence, Gaucher disease, it occurs due to the dysfunctional metabolism of sphingolipids and the deficiency of the lysosomal enzyme glucocerebrosidase. And because of this two factors, it results in the accumulation of glucocerebroside within the lysosomes. This enzyme, it functions to break down glucocerebroside into glucose and ceramide. But glucose macrophages, By macrophage, I mean any large mononuclear, highly phagocytic cells derived from monocytes. These macrophages, they have this accumulation of glucose cerebroside, and they appear engorged. This disease, it's a very rare genetic disease, but it's a very Gaucher disease. It's a common lysosomal storage disease. In canines, it occurs a lot in Sydney Silky Terrier dogs, and it can cause inflammation. It also results in organomegaly, which basically means enlarged organs. Thank you.